Hello students, this is a narration of slides 1 to 11 because slide number 12 contains questions of the nervous system content for this course. So I've decided to break up the information into small chunks. Hopefully that's helpful to you in terms of your learning, but it's also helpful because there is a lot of different pieces of information in this chapter, and so it's easier to learn small amounts at a time. So this first narration will focus on sort of give you a general overview of how the nervous system is organized and a little bit about how it functions. So we're going to cover, divisions of the nervous system. We'll take a look at the structure of a neuron, then what are the steps in the propagation of an action potential, sometimes could be called a nerve impulse, but a neuron and a nerve are not the same thing. A neuron is a nerve cell. Nerves are bundles of neurons. Once we have finished with that, this will be the end of the first narration, and then there will be other narrations to cover the remaining lecture content. So here we go. So the first big sort of anatomical division in the nervous system, so anatomy means structure, is the central nervous system versus the peripheral nervous system. So the central nervous system is made up of the brain, which is shown in the image here. So we have the brain and the spinal cord, which we all know the brain is protected by the cranium, and the spinal cord sort of passes through an opening in the vertebrae. So it's protected by the vertebrae. The peripheral nervous system is now made up of nerves, which I'm sure we all know we have. And this is sort of shown along the limbs, for example, nerves. So there are different names for nerves. For example, there are cranial nerves, and we won't be getting into the detail of those nerves. And then there are what are called spinal nerves. So as you can imagine, cranial nerves emerge, and they usually are associated with the head, the cranium, and then spinal nerves emerge from along the spinal cord. They actually travel between the vertebrae, and so they extend to all other parts of the body, to your fingertips, to the tips of your toes also. So this type of division, an anatomical division, is one way one could speak of the nervous system. On this slide here, what we see is a sort of diagram like a tree showing the nervous system overall. Then it branches into two parts, the central nervous system, the CNS, so that would be the brain and spinal cord, and the peripheral nervous system. And the peripheral nervous system really is all of the nerves and sensory structures that are outside of the brain and spinal cord. Now, the peripheral nervous system is further divided, and one of the reasons when we're studying anatomy and physiology that one of the reasons we create this sort of chart, this kind of diagram, and we break things down into smaller pieces, it's because we're trying to understand, we're trying to learn. So this is helpful in terms of our learning. But at the end of the day, the entire nervous system, both of these divisions have to function together. So the peripheral nervous system can be divided into a part that is actually voluntary. So it should not surprise you to see the voluntary control of skeletal muscle, because you learned last week that skeletal muscles require a nerve impulse in order for them to actually contract. So the voluntary control of skeletal muscle, that's referred to as the somatic division. So please pay attention to the words. We can use the words divisions sometimes when we're talking about the nervous system. Sometimes we can use the word regions. Sometimes we can use the word lobes, and they don't mean the same thing. So these are broad divisions for the nervous system. So the somatic division of the nervous system is voluntary. The autonomic, so this word sounds like automatic, but it's not automatic, it's autonomic. The autonomic division of the nervous system is involuntary. So this is again where we do not consciously control these responses. And so it's the involuntary control of glands and smooth muscle. So again, no surprise here, because you already learned that smooth muscle was involuntary. Now, the autonomic nervous system is further divided into two parts, the sympathetic and the parasympathetic system. So these two, the sympathetic and the parasympathetic, affect the same organs. So let's take the heart as an example. One of these, the sympathetic, can cause the heart rate to increase beyond the normal under certain circumstances. The parasympathetic will cause heart rate to go back to its normal rate. So normal heart rate is about 60 to 70 beats per minute. And if we're frightened, for example, or we're suddenly startled, we undergo a sympathetic response. And in that sympathetic response, we sometimes call this our fight or flight response. Our palms can get sweaty, heart rate increases, breathing rate increases. If you're at work and the fire alarm goes off, you don't know if it's a real fire or not, but you still have to leave the building. That would be a sympathetic response, because right away you're not thinking any longer of what you're doing. You're thinking about getting away from danger. So usually whenever we have to get away from danger or a situation that we perceive to be dangerous to us, our sympathetic system takes over. So what does the sympathetic system do? Some of the things I just mentioned. Heart rate to increase, blood pressure will increase, breathing rate will increase, muscles are primed for movement and running because you might have to get away. Pupils will dilate to allow you to see better, etc. Now, the opposite of that is where we would like to be most of the day, in the parasympathetic mode. This one is often called rest and digest. So it's called rest and digest because usually that's where we are, very relaxed, normal homeostasis. We're usually digesting our food then. Heart rate is normal, breathing rate is normal. Our pupils are just dilated enough for us to read whatever, we're looking at, whether it's small print or large print. But we are very calm. So the same organs are affected by the sympathetic and the parasympathetic system. But some of those organs go into a sort of overdrive because we're trying to get away from danger most often. Some organs in the sympathetic system are switched right off. Digestive is switched right off because the body does not want to expend energy on digesting food when your life may actually be in danger. So that's a look at sort of some of the broad divisions of the nervous system. Now, earlier I mentioned the word neuron. So on this next slide, we have now an image of a neuron. So you are expected to know how to draw, label a neuron, and you're also expected, this goes for all diagrams, and you're expected to know the function of the parts of a neuron. So let's take a look here. This is a cell, and it's a typical eukaryotic cell. So it has a nucleus there. This is the cell membrane, the plasma membrane, this outer part, as you would expect. One part of the plasma membrane is stretched out, as you can see here, and that part is called the axon. The axon continues, this is the length of the axon, and it ends here at these sort of, little branches. And at the very end of the branches are what are called the axon terminals. And that's actually where the neurotransmitters are stored. So we mentioned this when we were looking at how skeletal muscle functions. So in the nervous system, we have what is called neuron-to-neuron communication happening. So you can see very faintly there is another neuron here. And that neuron can pass information to this neuron that we're looking at, and then our neuron here can pass information to another neuron that is shown there. There's a small gap between two neurons, and that's referred to as a synapse. Just like there is a gap between the axon terminal of the neuron and the skeletal muscle when we were discussing the muscular system. Parts of the plasma membrane are also making these tree-like structures that are called dendrites. So the basic structure of a neuron isn't terribly complicated. Dendrites are responsible for bringing information to the cell body. So communication flows in one direction only in the nervous system. So information can come into the cell body. The cell body basically can make that decision whether that information will lead to an action potential. The action potential will start about there, and it will travel in this direction. So the action potential is always going towards the axon terminal. Now, while the dendrites all collect, so the reason they sort of have like lots of little fingers is because it allows them to cover a large surface area around, where the neuron is, so they can collect information and bring it into the cell body. Neurons have only one axon, but many dendrites. So this axon here conducts the information, or the nerve impulse, the action potential, I should say, away from the cell body. So dendrites bring information to the cell body. Axon takes information away from the cell body. Now within the cell here, all those structures that you normally associate with the cell, endoplasmic reticulum, Golgi bodies, that sort of thing, they're all present in here. They're not shown because they're not required for our discussion, really. So we're only focusing on the parts that we really need to know about. Now you're probably wondering, what is this sort of bluish covering that seems to be wrapped around the axon? But it's only wrapped around, at little areas. So there's a space between the wrapping. That wrapping is made of fatty tissue, and it's called myelin. So this is referred to as the myelin sheath, S-H-E-A-T-H. And that substance, myelin, is made by a special cell called an oligodendrocyte. So it's not made by the neuron. Myelin is made by an oligodendrocyte. Now, what is an oligodendrocyte? So within the nervous system, we have lots of neurons, but we also have some supporting cells. And there are actually more supporting cells in the nervous system than there are neurons, which may surprise you. So these supporting cells are called glial cells or neuroglia cells, and they are helper cells. They support, protect, nourish. I think of them as like parents. That's their job, is to make sure that these neurons remain very healthy. So that's what this specific oligodendrocyte does. There are other types of glial cells, that work like as a phagocyte. They clean up the environment where the neurons are in and make sure that it's always, healthy. So the question is, why do we only see myelin wrapped around the axon? And what is the purpose of these gaps? The gaps have a name, as you can see here. It's called the node of Ranvier. Well, when an action potential is actually moving, I'm just going to remove some of my markings to make this less busy here. Yes. So back to the action potential. So the next series of slides talks in specific detail how an action potential is generated. But what I wanted to say while I am on the slide is the action potential will start here, a place called the axon hillock, and it will travel along the axon. But when it gets to where there's myelin, it actually has to jump. So it jumps from node to node. Because if the action potential had to travel every little millimeter along the axon, then it would take a very long time to get to the axon terminal, and therefore communication in the entire nervous system would be really, really slow. And I think we all know how fast our nervous system can react. Because if I merely touch you with a sharp object or a hot object, you will respond right away. Milliseconds. So this type of conduction where the impulse jumps from node to node actually has a name. It's actually called saltatory conduction, but I don't think you have to know that. But that's how important it is. So we often say that myelin speeds up the transmission of the action potential, because the presence of myelin forces the action potential to jump from node to node rather than trying to travel every single little millimeter along this pathway. That would take very long. There is one disease where myelin is destroyed, and that disease is multiple sclerosis. So individuals who suffer from disease, unfortunately, have an immune system that is attacking their myelin. So when the myelin is removed, then their nervous system can't function efficiently. So we see this in the symptoms. They may want to reach and pick up a spoon or a pen. Movement. They know their brain is working just fine. They know what they're supposed to do, but they actually can't execute the movement. So myelin is not trivial, shall we say. So let's take a look then at how an action potential is generated. So now we have to talk about ions, and we have to talk about an electrical current. So when a neuron is not transmitting a nerve impulse, the action potential, right? That's the correct term, the action potential. It is called a neuron at rest. So that's very logical, right? And if you measure the inside of the neuron versus the outside of the neuron to see exactly, what kind of current do we have there, you will get a measurement that is called the resting potential. Now, because the measurement will be very small, we don't use a voltmeter. We measure in millivolts. This is just a smaller unit than volts. So usually if you're measuring electricity in a home or something, you can use a voltmeter and you'd measure in volts. We're going to be measuring in millivolts. I don't want you to get too concerned with the image that's here. The other images are more important for you to understand. So I'm going to read what's on the slide and emphasize a couple of points. So a neuron at rest, known as the resting potential, will have a negative charge on the inside of its cell. This is very important to know. At rest, the inside of the cell. So this is the phospholipid bilayer plasma membrane of a typical eukaryotic cell, because that's what our neuron is. So what we're saying is on the inside, all along the inside here of that membrane, sorry, it's down here, all inside the inside of the membrane, it is negative inside of the phospholipid bilayer. So this here would be the cytoplasm, if you want to think of it that way. That would be the cytoplasm there. And the outside is positive. So we have this difference in charge right away. Now, the two ions that are important in this discussion are sodium ions and potassium ions. So when a neuron receives an impulse from a sensory receptor, so we have sensory receptors all over our skin. If I poke you with a sharp object, there's a receptor that will pick up that information and basically send it to the brain. If you touch a hot object, or if you touch something that's very soft like velvet, right? The reason you can discriminate is because we have all these receptors. We have receptors for pain, for light touch, for deep touch, for temperature. So a variety of receptors are present all over the body and basically are just as part of our skin, the structure of our skin. So the two ions that are important are the sodium ions and potassium ions, and we're going to take another look on the next slide at where they're located. These ions will use a cell membrane channel protein. So if you remember from learning about the phospholipid bilayer, there were always proteins embedded within the bilayer, and those proteins can move ions. So those channel proteins help in this process. So let's see how we get an action potential. So here's our neuron at rest. Now we're showing you, so in this diagram, we're just looking at a small part, right? So there's our neuron, and this image here is of just a small part of the neuron. And so we see sodium ions are on the outside, potassium ions are on the inside of the membrane. So potassium ions, sodium ions. Now the question is, since they're both positive ions, how come we have negative charges and positive charges? And the reason for this difference is that there are always more sodium outside than there are potassium on the inside. And the sodium-potassium pump actually maintains So remember, perhaps from by 1150, you learn about the sodium-potassium pump and active transport. So the sodium-potassium pump maintains this kind of gradient that we're seeing here, where we always have more sodium on the outside and less potassium on the inside. So because we have less positive on the inside, that's why we have a negative charge. We can measure that negative charge on the inside. So the membrane is relatively impermeable to both ions. Think of it as the channels are kind of closed. So sodium can't go anywhere, potassium can't go anywhere when the neuron is at rest. Next, what has to happen is we have to have some sort of stimulus. Okay, so that the stimulus would be like touching you with a sharp object or the pin poking you. Okay, so that's a stimulus, and it has to be strong enough, because either you get a potential or you don't. It's an all or nothing situation. So if we get a stimulus that is strong enough, the first event is something called depolarization. So that's a word you have to know. Depolarization means that sodium ions are now allowed to enter into the membrane. And sodium ions enter, and what was negative, so this area was negative, now it becomes positive. Because more positive ions are coming into the inside. Potassium hasn't gone anywhere. So this is purely sodium. So depolarization is where sodium ions move inside, and that's the start of the action potential. Now the action potential has to travel. It has to travel along the length of the axon. So we have propagation then, right? This is propagation means something moves, slowly along a pathway. So this is what happens along the length of the axon. Sodium ions will come in. While that is happening, we get a situation where the action potential is traveling along the axon, and an event called repolarization is beginning to happen at the beginning. I'm calling it the beginning, but at where the action potential started, where the sodium ions first came in. Now we have an event where potassium ions diffuse out of the cell. So now potassium ions can move. So depolarization is sodium ions moving in. Repolarization is potassium ions moving out. So what you see here is that the action potential does not sort of happen like you switch on a switch and the entire axon just kind of lights up and changes in charges occur along the length all at once. That's not what happens. So there's this traveling of the action potential. So while one part is still undergoing depolarization, we have repolarization occurring at the start. So with repolarization, the charges are now going to be reversed again. Inside is going to become negative because more potassium is flowing out. So the channels that allow potassium out, they are open. And then repolarization will just continue along towards the end. So at rest, if you put this on a map, so here we've got time elapsed, And on this side, we've got the millivolts, right? What is the change? So if you measure the resting potential, that is always at about minus 70 millivolts, right? So the action potential hasn't started. The action potential begins when a stimulus triggers a neuron once past the threshold. So this here is what's called the threshold. That's when I said that the stimulus has to be strong enough. If it's not strong enough, you don't get an action potential. So once past the threshold, we get depolarization, which is where sodium gates. Sodium is allowed into move inside the cell, right? Changing the charge from negative to positive. So that's what this, and you can see that in terms of millivolts, it's increasing past 0 into the positive. The reason we have negative here is because you're measuring from inside to outside, and inside is negative. So we're getting that reversal where the inside becomes positive. That's depolarization. So it occurs along the length of the neuron. The impulse jumps from node to node. Next is repolarization, where potassium ions flow out of the cell. So for repolarization, which is on this slope here, you would expect it to sort of be opposite to depolarization because now potassium is flowing out and you're ending up with a negative charge on the inside. So we see the millivolts approaching. Anything below 0 would be negative, reflecting that. So the goal is to reestablish the original resting potential, because a neuron cannot fire another action potential until the first one that's got started is totally completed. So this is what allows neurons to relay messages within the central nervous system and the peripheral nervous system. Now, when the message gets to the end, see the end of the axon, we have the axon terminal, which again we learned. So this is now just showing you one axon terminal here. It's enlarged. It's like a knob at the end of the axon. And each of these little branches shown like a squiggly lines here, each one will have a knob like this. But these little balls that we see on the inside, those are sacs filled with the neurotransmitter. So eventually an action potential will reach the axon terminal of the neuron, and the synapse is a connection between the sending neuron. I would call this the sending neuron, right? Because the message is coming from it. And the neuron that is here, this is the receiving neuron. This is the one that wants to receive the message. So you see there's a little gap between them, just like we had a gap between the axon terminal, the neuron, and a skeletal muscle. The skeletal muscle would be here too. So that's what a synapse is. That sometimes is referred to as a synaptic cleft. The synapse is a connection between the neuron and its target cell, which can be another neuron or a muscle or gland or organ. So everything that you're learning here about the neuron applies when you're talking about a skeletal muscle needing a nerve impulse in order to know that it's supposed to contract. The axon terminals contain neurotransmitters, which are chemical messengers used by the nervous system. This is a really good word to learn, chemical messengers. The neurotransmitter diffuses across the synaptic cleft, which is the space between the neuron and the target cell, and binds to a receptor on the target cell. So we're saying a target cell, but it could be another neuron. This triggers the next action potential in the next neuron, so that would be the receiving neuron, or triggers the muscle to contract or the organ gland to function. So I hope this was helpful. On the next slide are these questions which you should do, and then you can check them in with me when whenever we have a meeting. So that's it for this series of slides. Take care.